Hello, everyone, and welcome to ACS Webinars, connecting you with the best and brightest minds in chemistry, live from Washington, D.C. I'm Michael David, and I will be your host for today's webinar, which we are proudly co-producing with the Joseph Priestley Society at the Science History Institute. Our moderator for today is Maria L. Maccecchini, the founder, president, and CEO of Annovis Bio. Dr. Maccecchini founded Annovis in May 2008 to better develop therapeutics for Alzheimer's, Parkinson's, and other neurodegenerative diseases. And with that, Maria, I will turn it over to you to begin today's presentation.
Thank you very much. Okay, I'm so happy to be here and be able to introduce a really interesting panel on artificial intelligence. Lately, we have heard a lot about artificial intelligence, and maybe not exactly in drug development, but in terms of faking people's voices, faking people's faces, faking people saying something, and faking writing. And of course, while that may help us in some ways, it's also a little disturbing. I don't think that in drug development, AI at this point is disturbing. It really helps us develop drugs faster, understanding them faster, and come up with drugs or compounds that have a better chance of not being toxic and being efficacious. So I'd like to introduce to you today a panel with three different aspects. One is it's artificial intelligence, how it was developed, how it can be used for a drug development, and how it can be used for population kinetics, how it can be used to actually help file an NDA. But that's Patrick, and I'll talk a little bit more about him in a minute. And then two CEOs of companies that are using artificial intelligence, in one case, to develop drugs for non-alcoholic fatty liver, and in the other case, for cancer. So let me first introduce the first speaker, who is Robert Foster. He is the CEO of Hepion. Hepion is developing a drug for fatty liver, non-alcoholic fatty liver. And he has used artificial intelligence, or maybe I should say, he has worked on drug development for over 30 years in several companies that he has founded and he has been CEO of. Lately, artificial intelligence has become more and more important in his drug development. Then there is Patrick Mayo. And Patrick has also been working in the field for a long time, but I think he started out as PK, pharmacokinetics, pop PK, population kinetics, so that you can actually predict how a drug will be absorbed by a person depending on their liver enzymes, their size, their sex, their brain, whatever. And then he has moved more specifically into drug development and to actually make drugs that fulfill a certain criterion. And he has worked with Robert now for multiple years in three different companies. Lastly, I'd like to introduce Panna Sharma. He is the CEO of Lantern Pharmaceuticals. Lantern Pharmaceuticals has several drugs for cancer. And interestingly enough, by using artificial intelligence, they have been able to decide which ones are good for the brain and which ones are good for blood cancers and which ones can be used for other cancers. So I am very happy to introduce Robert as the first speaker today. And Robert, start, please.
Thank you for the opportunity to give you a little background on our company and why we use AI. So, what I'd like to talk about is why exactly we use AI, and then Patrick Mayo will be talking about how we're using the AI. But I think right from the get go, we know that there's a lot of different diseases where it's hard to monitor how patients are doing. Maybe the disease is asymptomatic. So it is really hard to really get a strong handle on how these drugs are developed. And that's quite true when we look at liver disease and specifically, as Maria just mentioned, the non-alcoholic fatty liver disease, but also something more severe than what we call NAFLD, but which is called NASH, which is non-alcoholic steatohepatitis. We're actually in the midst of a healthcare crisis, and I know everybody was thinking more recently about the pandemic, But we do have a really big problem here with NASH and fatty liver disease. Just to put it into context, about 1/4 or even getting close to 1/3 of the global population has a fatty liver. If the fatty liver is left unchecked, it could develop into a more severe form of fatty liver disease, which is called NASH. Which, if you put it in the context of the United States, it affects up to about almost 20 million people in the U.S. that have NASH. So, for the people that are not familiar with NASH, if we leave NASH and we don't really treat it, it can develop or continue to develop into liver cancer, which of course is a very severe disease with this 5-year survival is less than less than 20%, and it affects a lot of people. So that is it's a really important healthcare crisis that we really need to go after. So what Hepion is doing is developing a drug for that. So what I'd like to do is to just kind of put everything into context and frame it so that when Patrick comes along, you'll see why we're using the AI. So part of the trick or the challenge that we have in NASH is that there's no current drugs used for the treatment of NASH, and there's no simple diagnosis. And in fact, for the earlier parts of the disease, people are asymptomatic. So how do you know when to use a drug for somebody who's asymptomatic, but if they're not treated, they can ultimately, get a severe form of disease and in fact, liver cancer or the need for even a liver transplant. So it's a really big challenge. And again, with an asymptomatic disease, there's no simple diagnostic and there's nothing that we can do in terms of trying to get our hands or arms around this disease. It is a big challenge. And I think this slide here, this third slide kind of encapsulates why we're trying to use AI. So on the y-axis of this graph, I've outlined a whole bunch of different drugs that are being tested in later stage clinical trials. Some of these drugs are still in development. Some are no longer in development because they just didn't work out. And if you look on the x-axis, that's the percent of the people that were in the clinical trials that responded. If you look at the dotted line with the black font, it says 16%. And what that number represents is it represents the response rate of people that have been taking a placebo. So no active drug, and you get about a 16% response rate. And that 16% could be attributed to lifestyle changes. For example, losing weight, exercising, these types of things that reduces liver fat. If you then look at all the different drugs that have been tested on the y-axis, again, these are later stage clinical trial drugs. At the high dose, which is the red dots on that particular graphic, at the high dose, we're only seeing response rates of just, sort of in a 25 or so percent, 24%, 25%. So the delta or the difference between the high dose drugs in development versus no drug or placebo is, only about 8%. So if you think about it, how do you develop a drug to treat such a severe form of liver disease, yet you're only having about an 8% pickup or benefit for people taking these drugs? This poses a significant challenge to people like us that are developing drugs. It also poses a big challenge to the regulatory agencies, as well as how do you commercialize these types of drugs. So for us, we've thought one of the things that we think can really help us, and not just us, but other people in this field that are trying to develop drugs, is we have to lean heavily on AI, because we have to do something about how do we identify people that are best can respond to these drugs. And again, there's a lot of people out there with this disease, but what we need to do is we need to pick the exact people that can take these drugs to benefit. Okay, so what we're trying to do now is, of course, develop a drug for NASH. As I mentioned, it's a multifactorial disease. Generally speaking, the NASH drugs have a poor response, and we have to do something about all of this. So we need to have, on the right-hand side of this page, we need to develop a drug to modify the disease. That goes without question. But hand in hand with that, we need to have a drug that can also, or we need to have an approach with our AI to also develop companion diagnostics to monitor how people are doing throughout the clinical trial process. And then ultimately, on the third point on the right-hand side of the page, is we need to develop a commercial strategy. Again, making sure that the drug is being used in the right population And if we can do that, then ultimately we can also seek the proper commercial strategy and also make sure that we get reimbursement for the drug that's being taken and used by patients. So what we have within our company is we have what I call it a pipeline within a product. So with one single drug, in this case, we call it Rancopelstat, we're not only positioning our drug development for NASH, as I mentioned, a severe liver disease that has a lot of fat in the liver, a lot of inflammation, and a lot of scarring of the liver. But there's other fibrotic diseases or scarring diseases that have scarring within the organ. One is the scarring of the lungs, which is IPF or idiopathic pulmonary fibrosis on the bottom of the page, and then renal fibrosis or kidney fibrosis, as well as cardiac fibrosis. And then you see there's a number of different cancer indications that we're also working on, one being liver cancer, the other one being multiple myeloma and prostate cancer. Those cancer indications are all quite early stage. And the leading, the leading development program that we have within Hepion is a phase 2B program for NASH, as well as a second phase 2 program for liver function. But again, because this is an asymptomatic disease for the most part, or certainly on the earlier stages, we really need to identify who is best suited to take our drug. And we also need to think, even though it's early in this stage of development, we need to be thinking about how to best position our drug for success commercially. So really, I think that's sort of what we're trying to do here. And in order for us to do that, what we need to do is we need to lead on the artificial intelligence. So for that, I typically lean very heavily on Patrick Mayo and his group, because they've been working on this for many, many years and have developed I think quite a unique approach to helping us in the clinical trial. So it's really a development using the AI for development, not so much discovering molecules, but in our case, it's on the development. So with that, maybe I'll just pass over the presentation to my colleague and good friend, Patrick Mayo.
Thank you very much to all the carbon life forms that are listening out there. And I'm here to talk about silicon-based life forms. And I don't think anybody's been in a position where they haven't been online, whether you've gone to YouTube and then all of a sudden starts saying, oh, you should watch all these things. Or you buy something from Amazon and you get, hey, other customers, they bought this. So if you've been something like that has happened, you've fallen prey to an unsupervised association type neural net of some variety. So you've been encountering AI right up firsthand and up close. I want to look at this little computer here in the corner. That's a computer that was in The Hitchhiker's Guide to the Galaxy. And the name of the computer was Deep Thought. And I want you to, this is a learning objective. I want you will, at the end of this, know what type of neural net you think that Deep Thought must be by the name of Deep Thought. And we'll just hold that in new neural nets as we move forward. So actually, let's just say, what is AI? Because there's all kinds of misinformation and things going on out there. And there is a strict definition. There's a group of scientists, and it might be an area of computer science, might be neuroscientists, mathematicians, statisticians, biologists, that are studying how machines can closely imitate human intelligence. And to them, unless you're actually talking about mimicking or even learning things the way a human being can, it's not AI. Now, that's a very, very strict and narrow definition. Now, where we're at, we're probably with a more working definition, which would be an area of computing science, statistics, mathematics. I come from, as you know, from a PK background that studies how machines can perform tasks that would normally require, and I put a sentient agent. You might say human being, but I put a sentient agent there for a reason. So what are we talking about right now? So you can define AI in all kinds of different ways, functionally and so forth. This is a very common one. And this is actually, if you go to his blog, Andrew Cochran, it's actually a very nice overview. But what we're typically talking about is what we would call artificial narrow intelligence. And I don't like this term. It says weak AI or narrow capability. And what I would suggest to you is, I don't like the term narrow. I prefer the term focused, because just like that Deep Thought computer, that computer, according to the story, was built to come up with the ultimate answer to the question of life, the universe, and everything. It came out and said that the answer was 42. People weren't too happy about that, but then they realized they didn't know the question. And that's what I would say to you. If you want to employ AI, machine learning, what is it you want it to do? What is your question? What's the reason for doing this? Because that's going to also stipulate what type of AI machine learning algorithms you're going to want to use. So I would argue very strenuously that it's not necessarily a weak AI, but it is a very focused AI. And so, with that, if you're talking about a machine learning algorithm that can parse through millions of discrete data points and then actually come up with hidden patterns for pattern recognition, or maybe I have very complex pharmacokinetics and I want to fit this with a complex algorithm, I don't think that's weak. I think that's pretty profoundly powerful. So that's the only thing I don't like about that designation. But the next step would be artificial general intelligence. And of course, we want even our narrow AI to not just fit our data, but to possibly be able to generalize to similar protein structures. So for PK, for PKPD, to enrich, to look at different patient types, things like that. But a bigger step would be an artificial general intelligence, also known as a strong AI, as general capabilities. Human beings tend to have more of a general capability. Is that in the future? Or is that now? I would argue it's probably a little bit more in the future, but some people might say, oh, we're getting pretty close. Now, the one that everyone gets all excited about in, movies are written and Elon Musk gets upset about is artificial superintelligence, the strong AI. And that's the one that becomes conscious and takes over the world kind of thing. Honestly, that's the machine that's really supposed to think more like human beings. It's transcendent capabilities. Is that possible? And I know some people think so. It's a really good argument to have over copious drinks and discuss deeply philosophical conundrums, but we're not going to go into that one today. So let's just look at this universe that's called artificial intelligence. And again, a very general definition would be a technique which enables machines to mimic human behavior. Might be as simple as doing a linear regression. I know how to do it. I can sit down, do it manually, or I can use my computer to do it. So it might be that it mimics the behavior that I had just to run a simple linear regression. But that actually fits more into the next little subset, which is machine learning. So that's a subset of AI techniques which allow statistical methods, and it enables machines to improve with experience. So that's interesting because I can actually have my AI training on a data set. And if I keep training that, it'll get better and better and better and better at fitting that. Now, if I don't actually test it on something or add in more data, it can overfit. And then you can get all kinds of bias because of that. I know this one set of data very, very well, but maybe my generalizability now becomes completely impugned, right? I want to go to the next one, and that's deep learning. And remember that computer's name was Deep Thought. And that implies that, we're thinking deep esoteric thoughts. Again, I have a philosophy degree, which I know just enough philosophy to really mess me up for life. But the deep learning is a subset of machine learning, which makes computation of multiple layer neural networks feasible. And the neural network, generally speaking, kind of mimics, kind of how a neuron, how you solve problems. But with a deep learning neural net, you have lots of layers. Whereas with a shallow one, you may only have one layer. I always argue that politicians probably are a shallow neural net and maybe scientists are deep, but I could be wrong about that. But maybe we'll let the AI sort that out. So, but based on that, you have some function, this deep learning. So I would argue to you that computer, a deep thought, is actually probably a deep learning or a deep learning neural net, or maybe a deep learning convoluted neural net. I'm not really sure which. Anyhow, so but what I want to make the real point is you can use this directly for drug discovery. And I've implied that already because we're living in an age where we have multiomics, and I can have genomics, transcriptomics, proteomics, lipidomics, metabolomics, and I can have that on all kinds of diseases. I can have it in all kinds of different protein structures, and I can use my AI machine learning to go probing through that to look for hidden patterns. So I might find new targets. I might find drug protein interactions. I can look at all these different things, and that's going to be more for the next speaker. But I also want to point out that if we're looking at the entirety of drug development, we can use AI at every step. And actually, AI machine learning is moving in and actually really helping us go through this massive amounts of information to solve problems much quicker. And maybe we don't have to do trial and error over and over and over again. My point is this, though. If you come up with a new molecule and you find a new target, it's got to be eliciting some change. Can you measure that? Can you turn that into a biomarker that you can now take, and you can actually take that all the way into preclinical and take that all the way into the clinic and then take that all the way so that you can actually help patients, not only with a new drug, but with a new marker that tells you the best way to use that drug. This, because a lot of times we would put people on drugs and it was almost trial and error. So we could use it in all these different aspects. What I'm going to focus on now is on the clinical side, clinical trial design, subject enrollment, patient, and monitoring. Okay, so this is kind of our AI power, and it's kind of a circle. Let's start the circle with external databases and real world data. That's at RWD. Now you're going to be, maybe you're trying to decide a new molecule for, you need a disease. Actually, regulatory agencies don't let you just develop a drug if there's not a disease. So you need information for the disease. You want to know what's going on. You want to know the pathophysiology, what proteins are involved, what biology is involved, and you need to know that very, very well. Now you may want to actually say, couldn't you actually, now what kind of molecules might work in that? So that's in that red box over there, that integrated system to tissue based on mechanistic models for mechanism of action and drug target. Again, as I said, when I decide that, maybe I've come up with a marker that tells me if I'm engaging that receptor. But could that also tell me something about disease progression? Am I actually hitting the right thing to make the patient better? So then you're going to go to the next thing in the wheel, which is bioanalytical data. I'm going to try to pull that together. Now, what we do, we also use AI machine learning all the way into the preclinical and before to predict if it's going to get into the tissue. Will it get to the site of action? Is there a possibility that the drug will actually engage the receptor? You take that and move into preclinical, and then I can actually even test what my hypothesis was out in test tubes and in cell culture. And then I move on and I take that further into the clinic. And what I'm ultimately trying to do, and I may do PK/PD at concentration effect, and we've actually integrated AI machine learning into that. But I also will do something called agent-based modeling, where I can actually then predict the outcome. Will this actually prevent somebody? Will it allow them to live longer? Will it improve the quality of their life? And ultimately, what I want to do with that is come up with a biomarker for the disease, a biomarker for the drug. That actually helps me enrich my sample to go into phase 3. So I don't have to put a bunch of non-responders. But ultimately, what I want to do is be able to hand the drug over and say, this person will respond, this person will not respond. This is new. We don't just put you on it for a month and say, come back if your leg falls off, or don't come back if you feel better, right? So it's a bit of an exaggeration, but what we're trying to do is move into the world of precision medication. So let's say you're a young chemist, you want to design a new drug and you'd like to use some of these approaches. Is there any way you can do it right now? And yes, there are. There's publicly available consortiums. This is the Therapeutics Data Common. And I'd urge you to go take a look at that. And if you go to the middle of it, you can see that they have like 7,000 different diseases. And you can actually start taking advantage of this AI machine ready data sets to design new molecules. From my perspective, I'm also very happy to see that they have things about bioavailability. So they have an ADMET thing. So I would heartily urge that you go take a look at this. And then let's just go on to the next slide, because I know Maria's going to I'm going to get the digital hook here pretty quick. And what I want to say is now if we just focus on drug discovery, that actually how you can actually use this to come up with brand new molecules and discover the disease. I want to now hand that over to the next speaker. I want to hand this over to Panna, who with his company Ladder, you can actually go to the public ones, but your one-stop shopping might be Lantern. So I'm just going to hand that over to him.
Pat, thank you very much. So as Pat mentioned, Lantern has been focused on developing a platform to develop and design drugs. We use it both for early stage discovery and development, where we've been able to take years and probably tens of millions of dollars off of our program development. We've launched 9 programs in 2 years at cost. So this is kind of what Pat alluded to as very narrow AI. We've developed a platform that brings in billions and billions of data points, a lot of that of our own, as you can see here. And on top of that, we have proprietary methods to tag that data, curate it, make better sense of it. A lot of that tagging and meta tagging now is automated. And then we run hundreds of algorithms to answer different types of questions. So our prediction success and the types of questions we ask it, we can range from very, very specific questions, very, very more complicated questions like mechanism of action in a certain tumor type, or where does the mechanism of action rest in the proposed mechanisms that are involved. our prediction success is kind of 80-90%. Actually, it's very much closer to 90+% now. And again, we use different types of patient records. We have over 150 drug tumor interactions modeled, meaning we know what the mechanism is for that class of drug, what's involved, what safety points are involved, et cetera. We were ranked one of the top end-to-end AI drug discovery companies. I won't talk a lot about our clinical programs because there's many and they hit it. I know you guys are logged in here today to learn really about how AI is changing the space of chemical development and chemical discovery. So we're ranked was one of the top programs based on the phase of our clinical program, as well as the types of questions we solve. As I mentioned, you can use and build reactomes to model mechanisms of action, look at mechanisms of action. That's very important because that leads us to maybe a very important part of being a pharma company, which is the next indication. We can use that to potentially uncover indications of interest for a compound or for a drug, or to really narrow in, pinpoint the indication where it's going to be most useful. Oftentimes, getting to that indication doesn't require just one compound, it requires several compounds. So we use different algorithms to look at what is the optimal drug combination, what's really synergistic versus purely additive. And we get some really good methods to extract things like SHAP features and scores and Bliss synergy scores, which we use quite a bit. And so for those of you looking at drug combinations, Bliss synergy scores used quite a bit. You'll see that in Therapeutic Data Commons. And then as Bob pointed out, one of the most important things for the trials to succeed is to have a signature. And oftentimes it takes a lot of time and money to develop these signatures of response. In my prior career, I developed signatures in a pretty large company for a lot of different big pharma companies. And the process took about 18 months, took tens of millions of dollars. And we can do that now for less than $1 million in 2 or 3 months. So things have changed quite a bit. Again, the key is the advanced algorithms. On top of collecting data, you actually have to tune these algorithms. You just can't pluck them off the street and think they're going to work. So we use a very important machine learning DevOps environment. So unlike a lot of companies that take AI off the shelf and use it, this is part and parcel of our everyday workflow. So we have a DevOps environment that manages and tunes all these algorithms constantly for a wide range of different examples. And again, we have both unsupervised as well as supervised. And we've actually filed patents on what we call ensemble algorithms, where we're taking pieces of different algorithms and bringing them together to come up with hopefully a more diverse and better answer to some of these questions. So how does this accelerate drug development? It allowed us to predict one of the major gene drivers for one of our drugs. We did this multiple times at a fraction of the cost that when we were first emerging as a company, we didn't have a lot of capital. So in silico methodologies were really important. And having that thesis around what the driver of being able to predict response is really important. And that also allowed us to identify an indication that we didn't think about. So today, glioblastoma, and one of the more notorious brain cancers, is a major part of our program that was overlooked by a lot of the biologists. But we saw by looking at the PTGR1 gene, and more importantly, the reactome around that GBMs would be very, very sensitive. When we took that to animal models and these unfortunate poor mice, it actually was true. And then when we also validated the PTGR1 hypothesis, we took it again to CRISPR gene editing to say, what if we remove that gene altogether or suppress it, quiet it down? You can see that the cells here where PTGR1 was knocked out remained very viable, whereas the ones that were overexpressing it, these horrible pancreatic cancer cells, we killed them off at about 100 or so nanomolar IC50, and by 200 nanomolar, they were all destroyed. So this was a great example of how an in silico multi-omic approach allowed us to go from idea to action. We filed for FDA orphan drug designation on the back of some good experimental work. And now we're launching a phase 1 trial in many of these cancers that express PTGR1 or have some of these DNA damage repair deficits. We do this not only for ourselves, but we do it for other companies. We did this for a company called Actuate. They did not have a platform, but they really needed to understand how to develop indications to expand their portfolio. We helped them do that by creating a neural network-based model for expanding their drug indications. And we use drug response data. We use patient survival data from phase 1 and 2 trials. We looked at a lot of in vitro data to really fine tune our assumptions about the biological mechanisms that were involved. And then we also use actual patient mutation data to say, is there something about the cancer and about the biology of specific cancers that we don't know that's going to get uncovered in the clinical setting? We then were able to do very important things like develop a biomarker connectivity map around the mechanism of action, and then also start modeling survival based on some of the patient data we got. We also look at very small things like blood-brain barrier permeability. If you go to Therapeutic Data Commons the next few weeks, you'll see our scores will be probably top ranked on the Therapeutic Data Common, kind of destroyed the existing numbers. And we did this by looking at these SMILE features only and creating about 4,000 to 5,000 features that represent most of the atomic properties. And a lot of the other Therapeutic Data Common models today use millions of parameters. And so we're using thousands of parameters. But what we also did in our algorithm around predicting blood-brain barrier permeability, which now I think we have the top 3 or 5 performing algorithms, is we actually created a meta algorithm that actually creates its own and selects its own features automatically. And that's really important because you can't always have this kind of constant supervision or human interaction. And you want these algorithms to always kind of live on their own. So again, we tested this for our own drugs, for lots of drugs. Here's what we predicted. Red is for our drug. Pink was the standard of care in GBM. And it said it had to be just as good as it. We went and actually did the in vitro experiments. Actually, it was just as good, maybe just slightly better. So this gave us a lot of comfort that our algorithms were great and working and giving us the right kind of information, but also saving us time and money. And we did this not only with our drug, but we can do it now for hundreds and hundreds of different drug classes. We also, like I said, we modeled which tumors are going to be most responsive. The proof is always in the pudding. When we went to we went into the lab to look at these 10 tumors that were predicted to be super responsive. They all were. And that was very exciting. But as Pat pointed out earlier, maybe Robert, is if I understand the commercial viability, 7 of these 10 tumors are resistant to PARP inhibitors. So that gives us a very good pathway to say, where to point our drug in terms of where is the clinical need, where are drugs failing today, and how can we point this? Typically, this would take years of work in the lab, and this took us probably less. So this is the power of AI, is to fine tune. We also published, I urge you guys to look at the publications we did with the National Cancer Institute in discovering a whole new mechanism with one of our drugs. And this was all done in silico and then validated in the lab. I know we have limited time, so I'm going to pause there and turn it back over to Maria.
Thank you very much for these very, very interesting 3 talks. I do want to point out that Panna's group just looked at one of our drugs that we do not know whether it's blood-brain barrier permeable or not. And supposedly it is, and it's actually being tested in rats as we speak. So in 2 or 3 weeks, I'll know how good his algorithm is. And now we have time for question and answers. So the first few are for Robert because he is the first speaker. And one question was that you said a third of the population is that the Western world? Is that the US? Who exactly has NASH?
Yeah, it's
Or
a
is
third.
that risk for
Actually,
NASH?
1/4 to 1/3 of the global population. So that's a lot of people. And if you look at, let's call it 25% of the global population has a fatty liver. 20%
But
of
after a
those,
certain age or at age 2?
pardon me,
At what age?
that's of the entire global population.
You mean
So
kids have NASH?
yeah, you can
Kids
have
have NASH.
kids can have NASH. It's not as common as somebody
Okay.
my
Okay.
age, but it's a quarter of the global population. So if you think about it, if you develop the more severe form of liver disease called NASH, there's about 20% of those people that have fatty liver that can go on to develop NASH. So it is a huge problem. And so many people, if you were just walking down the street and you just stop randomly, 1 in 4 people, and you were somehow able to look at their liver with your X-ray eyes or something, you'd realize that these people have fatty livers, but they have no idea that they have it. And so it's only later on when all of a sudden maybe they're feeling lethargic, they just don't feel quite right. Maybe they go to their family physician and they get checked out. They realize that something's going on in the liver. Maybe they'll get an ultrasound. Ultimately, maybe they even get a biopsy. And that's when they find out they have a problem. But by then, you've got scarring of the liver. So you've got this protein called collagen that's laying down throughout the liver, and you're starting to lose the function of the liver. So clearly, that's a little bit late in the game to start thinking about how do you treat these people, and that's the challenge. And as I showed, there's very, very poor response rates in clinical trials overall, and it's because we meaning drug developers working on liver disease, we don't really have the tools or the diagnostics to be able to find out who really needs these drugs in development. And that's where Pat and his group have come in, because they say, okay, well, given all these, thousands and thousands or hundreds of thousands of data points, we can identify, first of all, what the disease really is. And the disease is a little bit like moving goalposts, because it's changing as you get, more and more severe forms of disease. But ultimately, and you stack on top of that our drug or a combination of drugs, as Panna was talking about, where you use these combination approaches, then it's like, what is happening to the disease as you start putting in these medications? And we really do need AI to be able to sort that out. And that's probably one of the key reasons why, as it stands today, there is not a single FDA approved drug to treat this disease. So clearly it's a problem.
Yeah, the next question kind of follows that. When you showed the curve of responders, one drug had a 60% point. What drug was that?
I can't recall offhand. I don't have a slide in front of me. I can't recall offhand, but yeah, and that would be the high dose too. I imagine that would have been the high dose, but and it's also, when you look at those individual trials, it's hard to compare them. You're comparing a little bit of apples and oranges. So for example, it really boils down to inclusion and exclusion criteria a lot of times. And so for some of the trials, for example, what they'll do is they'll almost run like a pilot. They'll say, okay, if you can take this drug X and we see a certain amount of reduction in liver fat, then we will classify you as a responder. And if you're in one of those groups that you seem to respond, then you participate in the longer trial. But we're trying to avoid that approach by using the AI up front to be able to more accurately determine who's a responder without unnecessarily giving drugs to people that don't really need it.
Rob, even though this is a little further down, but it kind of follows in your thinking, a participant is asking why you are not using AI to identify a target for more effective drugs. And I'm not really sure I understood exactly what you're using AI for, but what he is saying that you're basically using AI to search for customers to fit the indication.
Yeah, no, I know. I would say that's probably a Pat type of question, but I would say, right now, Pat and I and some of our team are in Banff at a fibrosis conference. One thing that strikes me about listening to the talks is that every single speaker who seems to be stepping up is talking about a new target. So what it's telling me is that this is a really complex disease, and we don't really understand completely all of the targets. What we do understand, however, is our target, because we have a very specific mode of action related to the way proteins are folded. So we can gain a lot of information there, but we're not really looking for customers, to be able to work on these targets. I mean, it's really a way of understanding our drug. And then ultimately, when I sit down with Pat and his group, what amazes me is that Pat's basically telling me, and he can tell us in his own words, but you can run a phase 3 trial, for example, in silico, and then understand what your trial is going to likely provide at the end of the trial, which can cost tens and tens of millions of dollars. You run your trial in silico, and then by the time you get to your actual trial, you really know what you're doing, and you're saving a lot of money. And Panna also mentioned that you're saving a lot of time, you're saving effort and money, and you're increasing your probability of success.
It strikes
So
me
I
as
think that's our approach.
that maybe
Okay.
NASH could be maybe a handful of different molecularly classified diseases.
Exactly. Yeah.
Yeah.
I mean, actually, what I would sort of like to add to that, you say, well, we think we understand our target, but we've got all this information and we'd be foolhardy not to look at it and think that, well, maybe we're actually hitting other things. But I think, Panna, you hit it right on the head. NASH is very heterogeneous. And I honestly think there's a phenotype that looks like NASH and all kinds of subtypes underneath. And we realized very quickly when we wanted to do enrichment that there probably are, and that we did have to dip back in and look at the disease state itself and look at if there's any way to stratify the disease while we're also looking for, well, where does our drug work the best? Right. And so then that allows us to come up with a biomarker pattern to say, you have this type of disease and our drug is likely to work in that. We also can use that as a pharmacodynamic endpoint. But you're absolutely right. We haven't completely given up that. I'm open-minded enough to say, yes, we designed the drug to hit to do specific things, but it could be doing other things, and it behooves us to keep looking for those. Also, we didn't have the data set. I mean, the data is pouring in hourly, but not like in oncology. So we did have to dip in and we had to add with our own studies some of that information that we needed to do some of that target evaluation. But also we have a mechanism of action in mind. We can, as we go forward, I want to confirm that in the clinic, right? So that's why we're doing multi-omics in the clinic as well.
Yeah.
Help it all.
Things like for diseases like NASH, definitely things outside of, the typical genomic data is probably more important, transcriptome, enzyme, protein data.
Yeah, the transcriptome. I like the transcriptome, the reactome, because this is how your body's reacting to the disease, and then this is how your body's reacting to the drug. But we also have proteomics, lipidomics, and metabolomics.
Yeah.
Okay, the
Has
next
it, has,
question is for Pat. And this is on ADME PK and actually on POP PK. If you have limited data sets, like in the rare portion indication, how do you come up with a model? And here, some things are suggested. Can data augmentation, transfer learning, or domain knowledge be utilized to mitigate the impact of the limited data sets?
So, and I also saw what went by was, can you use synthetic data, right? And I would say yes. As a matter of fact, the FDA just they actually have a working group on AI and machine learning as it pertains on the clinical side. And so you actually can. So I can actually take and use my AI to generate patients, to generate synthetic data that I can actually use kind of to help, well, validate my model. Because this is a very similar process when we have in just POP PK, when we're looking for covariates. And for instance, what we do is kind of a bootstrapping thing where we simulate thousands of patients and then sample from them. Well, you use AI the same way. And so so that question was a very good question because absolutely, yes, you can. And the FDA is actually aware of that. And there was actually a study with Alzheimer's, and they actually used synthetic data. They created synthetic patients, and say, well, what would this patient have been like with or without the drug? And then they use that to augment, because as you know, it's like when you have we have orphan drug or orphan disease, you don't have a lot of patients. It's not a huge sample. And of course, you can generate a neural net on anything, but also the smaller your data set, the more difficult it is to make sure it's not a biased neural net.
yeah, there are issues with synthetic data also, but yeah, people are beginning to use synthetic data populations. I think it's a lot easier to try to use other types of models, but synthetic patient data, because of the very nature of synthetic data, you might end up with a lot of noise, a lot of meaningless noise, because you can create synthetic images, synthetic genomic profiles, synthetic reactions to first line therapies. But because they aren't governed by the real world, you don't know what's going to come out of it. It can just be a lot of junk. And so if you're going to create algorithms based on synthetic data, I think it can be really, really challenging. Now, you can insert human intervention into synthetic data, at least for cancer. But then you get into the bias of the human touching all that data to decide what is relevant and not relevant. It's a real problem, synthetic data for some of our models. We think about it in a very specific areas like blood-brain barrier permeability for compounds, because you can create in silico compounds, types of compounds. When it comes to real world, like companion diagnostic type development, I would steer away from use there today.
Well, I would say though, what you do is kind of what we always do is, let's say you want to try it. Well, then as you go forward, you'd want to test it. So as long as you don't just kind of let it go and say, I'm putting all my faith in this synthetic data that I've created, you actually then do some sparse sampling to see if you can refine it. And is your synthetic data really a good representation of what you've got? But I see what you're saying, Pat. It makes a lot of sense. The fact is, the FDA is actually open to that, but of course, then validation becomes a really critical thing. And once you're on the clinical side of things, it's validation, 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 right? So, but I agree. It's not like I would do that without a lot of checks and balances.
Yeah, that's correct. Yeah. Good point.
Okay, the next question are probably more for Panna and maybe Pat. So one question is, in silico calculation to prodrug design to change the permeability properties. Can you comment on that?
I don't think I understand the question, Maria.
Well, I don't either, to be honest, because it continues. So in silico calculation to prodrug design to change the permeability properties of a prodrug to enable liposome formulation. I don't understand how a prodrug has anything to do with liposome formulation, but that's why I gave you the first part.
Sounds like someone's working on a specific problem, is what it sounds like to
Yes,
me.
exactly. Okay, we need more information to answer that question. And then whether AI would actually supersede combinatorial chemistry. Do you think that's going to happen, or has it already happened?
I think
I would just, I would
be part and
sorry, Pat.
parcel of using it. I think the field just begins to change. It's another tool. So the pace at which combinatorial and medicinal chemistry needs to occur, it needs to be faster and cheaper. And so I don't think this field goes away. I think it just evolves to being able to leverage machine learning.
Yeah, I agree with Panna. It's like all these things, even the way when we put them in with PBPK and POP-PK, and you can even do patient responsiveness models in POP-PK. It's not like I use it to replace everything I've done, but it's a tool. And it also allows me, especially if I got multiple iterative stuff, if I've got and also if I'm trying to go through this massive amounts of data, if I sit there looking at it bleary eyed, I can drink all the coffee I want. I'm going to pick what I like, miss what I don't. The computers they don't get tired. They follow the algorithm and they do the work that I can't do, even staying up all night. I'm actually, I look old, but I'm like,
Well, I would just add, Maria, that, years ago, I toured through some of the big European drug company manufacturing plants. I'm sure a number of people here have as well. And, I'd go into the chemistry labs and there was just, basically robots essentially that were doing the old mix, beat, heat, what is it? Mix, beat and heat approach where they just have all these, essentially funnels and lines that just lead into, building these molecules. And then they would do this high throughput screening. And it's really just the shotgun approach. Like you just throw everything together and hope you get a hit in the high throughput screening. But I think where AI comes in is it's pulling together knowledge of the disease first, the targets, and then trying to, from a drug development standpoint, building these molecules that will hit these targets. So there's a lot more, I think I would say a lot more thinking that's gone into this. And it's just an evolution, from what I think many of the drug companies were doing at the time and maybe still are.
Yeah. The next question is a question I have myself. How can AI be used to predict the outcomes of clinical trials?
how did you want me to go? Because I've got thoughts on that for sure.
Okay,
Um,
please.
I mean, he alluded to that as well. I mean, so what you want to be able to put in your data sets is, so let's say drug trials fail. Well, that's still valuable information. So you've actually got some information on what happened, what didn't go happen. And you always have responders and non-responders. But for instance, in NASH, you got to look at fibrosis because that's the one that's linked most intimately with mortality. And ultimately, people die of cardiovascular disease. So I'd like to link my biomarker early on with that kind of a tangible clinical outcome. As a matter of fact, that's the biomarker you want. You want to link your biomarkers to clinical outcomes. And then so that you can hopscotch a little bit to what's gone before. So that's why you're always going through. And I never look at a drug trial, a drug that's failed as a complete failure because it gives us a lot of information. I think Panna might agree with that. And so then that allows me to say, okay, I've changed this, that affects this. And then now I know that that's going to affect these downstream events. And now I can link that to clinical outcome. The other is that I go through my clinical trials. Of course, my phase 3 is where I really it all should be coming together. I should be able to link that. What I ultimately want to do though, is not just have something that helps me get into phase 3. I want to make a drug that helps people. And so that's why you absolutely need to link these kind of biomarker panels to relevant clinical outcomes.
Okay, thank you. Here is a question on using AI for toxicity adverse events. How well is that going?
I think safety and tox predictions for unknown classes of molecules is really good right now. So if you go to therapeutic data commons, you'll see a lot of ADME and tox data across lots of different molecules. For new classes of molecules and for combinations, it becomes more tricky. For pediatric cases, it becomes even more challenging. For patients who have comorbidities, so for example, they have a blood-brain barrier that's more leaky than expected, or they have cardiac and heart issues, or they have existing issues with liver function. Those are all become very challenging. Those are hard to predict. And oftentimes they're unknown because these patients, at least from a cancer perspective, will go into the primary diagnosis and challenge of cancer, but they may have underlying issues that cause adverse events that are unknown at the time because of other comorbidities or other situations. But in general, the algorithms that exist now for known classes of molecules are really very good. When we get to break down in large molecules or biologics, they become more problematic because you have injection site problems, you have issues with immunogenicity of certain antibodies or peptides, and that becomes harder to model and understand. But for small molecules, I would say a great starting point.
Since we're getting close to 2 o'clock, I wanted to use 2 more questions. One candidate or one participant is asking whether you can suggest some literature, and if you could please do that and maybe send it to ACS so ACS can distribute. And the next question is a closing question. How do you predict AI in 25 years? And that I think we can give to all the speakers because it really closes off the panel and it ties out what we're doing today and how we see the future go in the next 25 years. So please, Robert, start.
Boy, 25 years. Well, with any luck, the AI will be so good that I won't have a job. So, um, no, I honestly, I have no idea. I'm old enough to know that, when I took pharmacology, many years back, we always talked about the lock and the key, so the key being the drug and the lock being the target or the receptor. That's so far gone out the window, in really not too many years. So where do we go beyond that, in 25 years with AI? I think, we're just cracking it open now. And we still have a lot of different diseases that are still super challenging, Alzheimer's being one. And there's so many other ones that we're just not there yet, especially neurodegenerative types of diseases. But the more and more we can learn from, if you want to call it like liquid biopsies and AI, the more I think we can learn about these diseases, the more I think we can identify molecules that target these things. So in 25 years, I think the technology is going to be such that we're going to have a lot of cures for a lot of diseases. And ultimately, people could be living a lot longer than they currently are with a lot of better quality of life. That's where I would see it.
At-
So, well, first of all, the first part you said, there actually is a reference in my slides. It's a very nice overview paper. I'd recommend people go look for it. They even give you some places to go. So if you want to start tinkering around with some AI things for chemistry and things, it's right there in that paper. So I'd look at the slides. There's a reference right there already. As far as where's AI going to be in 25 years, I wish I could hand that over to my colleagues who are both younger and smarter than me, Carolyn Zhao and Aaron Foster. But what I would say is, I think that we're just touching the tip of the iceberg, especially on the clinical side. I think that what's going to happen is all that high throughput screening, things like that. Panna's already shown this, is that we're going to be able to use AI to really, we're going to understand, we'll be able to sift through more information quicker to save ourselves a lot of time, a lot of cost. I think also there's going to be a lot of, we always have, we'll try with, oh, this is the great way to go. This is the terrible. And it'll settle down so that we'll have very, I think it'll become hard and fast. Some of the tools, we'll use them in all of our disciplines. It will become like, how you go to the weather map every night and this is the weather, but the model said, the projection was. And I think that more and more that's going to be something that's going to be, it's becoming an integral part of this whole process. And I think that, we'll discover what's best and throw away the stuff that isn't. And it's always going to be evolving. That's what I would predict.
Panna.
I think by 2048 or 2050, you're going to have an avalanche of precision medicines in cancer. Just look at where cancer was in 1998 versus today, when the pace of change is only becoming more rapid. I think that the bottleneck will always be clinical application. I think clinicians will be slow to adopt new personalized approaches. I think you're going to create cocktails for patients out of the gate. I think people even will have early symptoms of cancer and we'll be able to give prophylactic drugs early on and change those drugs. Today, we have about 100 drugs, about 70 to 80, which are kind of quasi-targeted. That's less than the number of cancers. So if we have about 500 to 1,000 different cancers that we know, we need at least 2,000, 3,000 drugs. So AI is definitely going to be needed. Clinicians are going to be a bottleneck. I think in advanced countries, you're going to have tremendous care. I think we're going to wipe out a lot of cancers and be able to better manage them. But I think in the second and third world, you're going to have a lot of have-nots. I think this is going to come at a tremendous cost to the healthcare system and a tremendous merging of tech and healthcare. So I think it's going to be a totally new horizon in the future. I think these things that we see where people like literally walk in, get totally scanned from the transcriptome and their genome and their proteome, and then say, these are the things you should eat, these are the drugs you can take. These are the conditions you're going to come down with. Here's some early markers to those conditions. Take these things prophylactically, take these things therapeutically. I think we're going to see that era, but I think it's going to be available for the wealthy. And I think it's going to be challenging to get that to drip down because of bottlenecks in clinical acceptance.
Well, thank you very much. We have come to the end of our panel, and I really appreciate having learned so much about AI. And we'll see where we are in 25 years. But I agree with Panna that especially in cancer, we have made enormous progress. And now we have to slowly catch up in other areas. Thank you very much.
Good afternoon. Thank you for joining us today. We appreciate your participation. Located in historic section of Philadelphia, the Science History Institute tells the story of the chemical, material, and life science sectors to both professional and audiences and the general public. Feel free to visit the museum and library in addition to the public space. We also put on a large number of both in-person and virtual programs, and we especially appreciate our partnership with the American Chemical Society that you participated with today. So please visit the website sciencehistory.org to see more of what's in the collections and what's available from the Institute. Thank you.
Thank you for watching this presentation. ACS Webinars is provided as a service by the American Chemical Society as your professional source for live weekly discussions and presentations that connect you with subject matter experts and global thought leaders concerning today's relevant professional issues in the chemical sciences, management, and business.